Um, so let me introduce our two clinician researchers who will be addressing us. Dr. Iosifescu is the Director of Clinical Research at the Nathan Kline Research Institute, an Associate Professor of Psychiatry at NYU School of Medicine. In his research, he uses neuroimaging and neurophysiology techniques to elevate structural, biochemical, and functional brain abnormalities in mood and cognitive disorders and their impact on clinical treatment. He received a 1.8 million grant through the Part the Cloud Gates grant. And based on his previous research, he saw that mitochondria may face damage early on in Alzheimer's disease, but near infrared light may help restore some function. So Dr. Iosifescu will be updating us on his research. Then he will be followed by Dr. Andrew Varga, who's an associate professor in the Mount Sinai Integrative Sleep Center in the Department of Medicine at the Icahn School of Medicine. Dr. Varga received $150,000 through the 2018 Alzheimer's Association research grant to study if sleep disruption leads to more rapid buildup of abnormal tau protein in the brain. And so I just want to say that these are really accomplished clinician researchers. Their bios are much longer than I've been able to condense in the short amount of time, but I thank them so much for participating in this event and sharing their knowledge and expertise. So I'm going to present on a general overview at advancing the science and where we stand, and make sure that we are all on the same page in terms of Alzheimer's disease and some of the terminology we use. So I'm briefly going to give an update on the landscape, talk about the highlights in early detection and diagnosis, and share with you the three reasons I have for optimism in terms of where we are in advances in clinical trials, treatments, and lifestyle interventions. So our mission at the Alzheimer's Association is that we are a worldwide voluntary health organization that is dedicated to Alzheimer's care, support, and research. And our mission is to lead the way to end Alzheimer's and all other dementias by accelerating global research, driving risk reduction, and early detection. So this presentation is really for the lay audience, because many researchers are very aware of where we stand in the science, the Alzheimer's Association's role, but we've realized that we haven't articulated for the lay public where we actually stand and what's really exciting in terms of the state of research. So I hope in my very brief remarks to give you a little bit of an overview. So we know that there's a huge impact of Alzheimer's disease, more than 6 million people affected. 1 in 3 seniors dies of Alzheimer's disease, and that is more people than affected by breast and prostate cancer combined. Alzheimer's disease is the most expensive disease in America in terms of healthcare costs at $355 billion. And that's not accounting for the $257 billion that caregivers spend providing informal caregiving. We know that there are huge disparities in terms of who is diagnosed with Alzheimer's. We know that African Americans and Latinos are diagnosed at higher rates. And some of this is driven by risk patterns, but also by barriers in the health systems, most notably discrimination. So when we talk, we often use the terms Alzheimer's disease and dementia interchangeably, but I just want to make sure that I clarify that they are not the same thing. When we talk about dementia, we're talking about a collection of symptoms like changes to memory, thinking, or behavior. More than 100 different diseases can contribute to this syndrome called dementia. And we name the dementia by what is the underlying disease that is driving the biological changes. So we have Alzheimer's disease that is the most common cause. but you can also have dementia that's caused by disease of the small blood vessels like vascular, Lewy body dementia, frontotemporal dementia. We very often see mixed dementia when two underlying diseases are driving the clinical picture. And this is why it's so critical that if you or your family members are seeing changes in cognitive status, that you get a full clinical evaluation so a medical provider can do that full clinical evaluation to understand what the underlying cause is. So Alzheimer's disease is a continuum. So we know that many times that people are asymptomatic, then they can develop mild cognitive impairment, which is an early stage of cognitive loss. But in that stage, the person retains most of their function. It is only when the changes to memory, thinking, and behavior interfere in daily life that we call something dementia. So we know that if we interrupt the development of MCI, we can develop and prevent the person going on to dementia. So we know that before there is a cognitive deficit, that there are underlying biological changes that happen in the brain. And many of these biomarkers change 20 years or more before symptoms appear in the brain of somebody who has Alzheimer's disease. So the prevailing hypotheses that we have in Alzheimer's disease is that the brain is composed of millions and millions of neurons that perform specific functions. In Alzheimer's disease, we believe that there is a buildup of beta amyloid, which is a protein outside of cells, and then a buildup of another protein called tau within cells that causes neurofibrillary tangles. Those two buildups cause the cells to die, and cells die first in areas of the brain that deal with short-term memory, but then affect other areas of the brain like the frontal lobe that affect a person's ability to plan, to reason, their personality, but also then goes to other parts of the brain like the motor cortex and affect people's ability to move, but also their ability to recognize faces. It affects their personality, et cetera. And so that is what affects the clinical picture that we see. So what is incredibly exciting is in these years of research, we've learned about these clinical biomarkers like beta amyloid and tau. In terms of where we stand in terms of FDA approved therapies, prior to 2021, all the therapies that we had available were focused on symptoms, and we're really trying to address some of the cognitive deficits we see by improving certain brain chemicals. In 2021, for the first time, we had a medication that was approved called aducanumab, or its trade name Aduhelm, that it's the first in its class that has the potential to have clinical benefit because it actually targets amyloid and clears amyloid. So we know that multiple things impact a person's risk for having Alzheimer's disease. We have non-modifiable risk factors like age, genetics, race, ethnicity, which we can't really do anything about. But then we also have modifiable risk factors like lifestyle factors, cardiovascular health, physical activity, diet, sleep, social and cognitive engagement. And it is so important to understand what modifies the risk, the mechanisms of risk, so that we're studying dementia risk from all angles, and so that we can develop more efficient ways to intervene to prevent Alzheimer's disease and dementia, and to develop better ways of diagnosing and treating. So behind the various research efforts fueled by the Alzheimer's Association, advancements are always happening. Now, many times in our clickbaity news cycle, you may not be aware of that, but not just in one area, but on every front line across disciplines, fields, industries, milestones are being achieved. So now I want to share with you some points of optimism. So my number one point of optimism is that today we have unprecedented levels of funding for Alzheimer's research from both the government and philanthropic sources. So when we first started our advocacy in 2011, and we used regular people like you as advocates to talk to our elected officials about the importance of research. And when we started this work in 2011, research at the federal level for Alzheimer's disease was $448 million. Today in 2022, it's projected to be $3.4 billion. And those research dollars translate into more researchers being able to study this, more postdocs, more undergraduates, more of the brightest minds in this country focusing on this problem, and more questions asked and answered about dementia science. So that's one area to be incredibly optimistic about. And then on the philanthropic side, the Alzheimer's Association is the largest nonprofit in terms of Alzheimer's and dementia research in the world. And so we have more than $265 million currently invested in over 750 best-in-class research projects all across the globe in more than 39 countries. And as an organization, we know that there are bright minds all across the globe that are asking and answering questions. And as an organization, we want to make sure that we are funding areas that sometimes the federal government cannot. And so that we accelerate the global research community through our grant-making portfolio. So my point of optimism number two is that there are more investigational drugs in the pipeline for Alzheimer's treatment than there have ever been before in the history of Alzheimer's. And I think it's instructive to take you a little bit through the process of how we get to the fact that a pharmaceutical company could be submitting a drug for FDA review to see if the drug will be approved. So first, research starts really at the bench, where there are scientists looking at laboratory experiments, seeing if they can discover mechanisms in animal models and test these cellular and animal models to understand a little bit more on the biological mechanisms. So research can be in this preclinical phase for years to decades. Then once there's an investigational agent that's discovered, it's trying to prove whether that works in human volunteers. And so that's phase one research, which can take months or sometimes years, but typically it's maybe tens of volunteers, sometimes 100. In this first phase, you're particularly looking at risks and side effects and getting initial information on dosage, but really proving the concept in human volunteers. Then phase two research, again, can take months to years. Then you're testing this investigational agent or drug in more volunteers, typically hundreds of volunteers. And in this phase, you're getting more information about safety and dosage, and again, more information about efficacy. Once you are in phase two and those studies are successful, you typically advance to phase three that can then also take years and decades. Typically in phase three, there's 2 studies that are done at the same time, randomized controlled clinical trials. In this stage now, usually we're testing thousands of volunteers, and this is really where the chief evidence is for an investigational agent. By this time, you typically know the dose, you know the safety profile, but now in thousands of people, you're seeing how clinically effective it is in terms of the trial endpoints. And it's only at this point, once there is the data from all these phases, that a pharmaceutical company then can go to the FDA for review, and then the FDA makes its decision to approve a medication or diagnostic device or not. So what's exciting is we see that there's currently about 131 investigational agents in phase 1, 190 and 67 in phase 3. And so this is incredibly exciting. And often in the news, when you hear about a negative study, they're telling you about one study, but they're not telling you that there's more than 388 other drugs that are in the pipeline. We know that it is incredibly important that at all stages of this development that we need to have diverse volunteers represented from diverse ethnic groups to make sure that the drugs being developed work in all populations. And the Alzheimer's Association has the TrialMatch program to connect healthy volunteers and also people with dementia to researchers who are working on more effective ways to prevent, diagnose, and treat Alzheimer's disease. One of the things that's particularly exciting is after the approval of Aduhelm, it seems likely that 2 or 3 pharmaceutical companies will be bringing forward their agents in 2022 for FDA review. We think that's the case for 2 new drugs that are in the pipeline. So we look forward that 2023 and 2024 will hopefully be exciting years that possibly will have 2 new drugs on the market that are focused on beta amyloid clearing, and that also have some strong signals in terms of improving cognitive function. So one of the things that many times there is a criticism of is that research is too focused on amyloid beta and tau, but there are many other underlying mechanisms of disease like neuroinflammation, vascular stress, oxidative stress, all these other things that are shown on the slide, but want to assure you that in terms of the Alzheimer's Association funding portfolio, we are supporting scientists that look at all these mechanisms because we recognize that we need to have more shots on goals, more questions asked and answered, so that we can develop more effective ways of preventing, detecting, and treating Alzheimer's disease. So one of the ways that we do that, which is a new initiative that we have, which is called Part the Cloud, which is started in partnership with the Alzheimer's Association and Mickey Hogue, who's a philanthropist in the Bay Area, and she's been joined by Bill Gates in this Part the Cloud initiative. And Part the Cloud is really about accelerating the piece of research so that we shorten the timeline from when there is something at the bench to the bedside. We have more than 59 clinical trials in the pipeline right now, 60 million funded, and you'll hear from Dr. Iosifescu, who's been funded also under the Part the Cloud initiative. And this is really exciting in terms of diversifying the clinical trials pipeline. Our third point is we are learning more than we ever have before about preventing dementia. And as a public health physician, this is what makes me excited because we have a recipe potentially that's on the horizon for how we prevent dementia. So initial information about this, is through the Sprint Mind study, which was the first study of its kind to demonstrate reduction of new cases of cognitive impairment. And I told you before, if we can reduce mild cognitive impairment, we can also hopefully reduce people who enter into dementia. And so this study looked at tight blood pressure control, keeping people at 120 systolic, the top number, versus people at 140. And it was found that if we kept people at 120 systolic, we could reduce mild cognitive impairment and dementia. And so this research has immediate life-saving potential, which is exciting. But then there's also other research that supports the idea that multimodal interventions that take into account physical activity, healthy diet, cognitive and social stimulation can reduce dementia and mild cognitive impairment. First, this comes out of studies in Finland, the Finnish geriatric study that showed that a multimodal intervention that included tight control of cardiovascular risk factors, diet, exercise, social cognitive engagement could reduce mild cognitive impairment and dementia. And so now in the United States, the Alzheimer's Association is trying to replicate this study because we understand that in the United States, we have a very diverse population. Our diets are different. And so we're doing a landmark randomized controlled clinical trial in 5 sites across the United States with 2,000 participants. And they'll be randomized to 2 groups. Either one group gets just a self-study in terms of how to modify their risk factors versus another group that gets more detailed support. We'll be following them over 2 years. And so if this intervention proves effective, this study will really lead the way in developing an accessible community-based program for prevention. In the same way that we know for cardiovascular disease, there are particular things that you can reduce your risk of cardiovascular disease. We hope that the results of this study will give us that specific recipe for how we prevent dementia. And what's exciting about this study is that these are regular American people that are couch potatoes and have a sedentary lifestyle like you and me. So that's incredibly promising. So I hope I've given you an overview on why it is an exciting time in research, how the Alzheimer's Association is a global leader for Alzheimer's and dementia study, that we have new tools for detection and diagnosis in the pipeline, that we have a growing diversity of therapies under investigation, and we have some strategies to promote diverse participation. And so all in all, there is hope and research. Now, I mentioned to you that we have TrialMatch, which connects researchers with both healthy volunteers and people who have dementia. And healthy volunteers are, could you mute? I'm hearing somebody come through. So participation in clinical trials is critical for our ability to develop new ways of preventing, detecting, and treating Alzheimer's disease. And there are all different types of studies in TrialMatch, from online studies to behavioral studies to clinical trials. And the database is constantly being updated. So if you're interested, go to alz.org/TrialMatch to learn more, or call our 800 number, which many of you use to register. And then finally, plug, we have an app in the App Store and in the Play Store that gives you all the latest updates in science and research, who we fund. And I encourage you to download that so that you get credible and vetted research information. So I want to thank you so much for listening. And now I want to stop sharing and turn it over to Dr. Iosifescu. Again, I encourage everybody to mute themselves, and if you have questions as Dr. Iosifescu is presenting, please put them in the chat. Dr. Iosifescu.
Thank you so much for giving me the option to present for this audience, and even more for the opportunity to have our study funded by the Alzheimer's Association and the Part the Cloud program. So I'm going to take the next few minutes just to introduce this very exciting, at least in my mind, very exciting research that we're doing. And it has to do with photobiomodulation. So this is using near-infrared lasers to essentially affect brain functioning. My name is Dan Iosifescu. I'm an associate professor of psychiatry at NYU School of Medicine, and I also direct the clinical research division at the affiliated Nathan Klein Institute. And I want to start this by introducing you to this concept of mitochondrial dysfunction. Essentially, there's a large body of research that suggests that mitochondria, which are the parts of the cell that produce energy, are not functioning well enough in people with Alzheimer's or early Alzheimer's disease. And if you listened earlier to Dr. Tavares' presentation, you heard about these very well-defined abnormalities of the accumulations of the amyloid and tau, either outside or inside the brain cells. And there's a very clear link between that and the mitochondria functioning. We don't really know which one is the chicken and which one is the egg. So on one hand, these accumulations of these proteins, both outside and inside the cell, actually do also impact the mitochondria, and these deposits actually bind to the mitochondria and make them start working more poorly. But it's also the other way around, that if either through some genetic pre-existing abnormalities, mitochondrial DNA is inherited primarily from mothers, or other types of mutations that lead to some minor mitochondria dysregulation, the clearing of these abnormal proteins actually requires a lot of energy. And if the mitochondria are not working well enough, essentially both the amyloid and the tau start to accumulate more. So it's essentially a vicious circle where the deposits influence mitochondria in a bad way, and mitochondria is necessary to deal with this problem. So what ends up happening, without really resolving which one is the chicken and which one is the egg, is that the more mitochondrial dysfunction people have, the more of a deficit in their cognitive abilities, memory, attention, executive planning, to the point where, as you can see in this graph from a recent paper, when mitochondria function is relatively normal, there's just normal brain aging. And that actually allows for some minor level of mitochondrial dysregulation. But then once the mitochondrial function decreases enough, one starts having either a mild cognitive impairment or at the more extreme range, Alzheimer's disease. So the question now is, can we do something about it? And whether we influence the original cause of the problem or just some other mechanism that can help clear the problem, enhancing mitochondria would be a good thing. It's clear based on this research. So what can be done to enhance mitochondrial functioning? The first studies were done with some natural supplements. So there's a list here of things that are supposed and shown in some animal data to have some positive effect on mitochondria. The problem is that none of them is extremely powerful. And people have tried to use combinations of them to make them more powerful, but it's not really clear that this is good enough. And there's ways in which you can measure mitochondrial functioning in living people. And most of the studies that use these supplements were not very exciting in terms of how efficacious they were. But in parallel, we actually stumbled upon a completely different strategy. So before mitochondria became a part of our complex cells, they were cells on their own that eventually were captured and started becoming symbiotic in these larger eukaryotic cells. And from that time, when the mitochondria were functioning on their own, they retained some ability to convert light into energy. So there's this part of the mitochondria, which is in the mitochondria wall. It's called the cytochrome C oxidase. It's an enzyme which is very important in the flow of electrons in mitochondria and their primary role, which is to produce energy. And this cytochrome C oxidase, if one shines light upon it at very specific light waves, is going to change and become more active, and the entire function of the mitochondria increases based on that process. This has been very well shown to work in cell cultures and in animal models. The problem to using this in the brain is that one has to shine the light through the skin and the bone, and a lot of the light is getting dissipated in that process. So without making holes in people's heads, which is not something that I would get a lot of volunteers for, it's a little bit harder to get the light in. But still, we're able to get a significant amount of laser light going through the bone into the brain and affect brain functioning in a positive way. Our early studies were done with patients with depression, and we found some positive effects in depression. And then there were some other groups that found in healthy volunteers improvements of cognitive functioning. There were also some very early studies in just a few patients with Alzheimer's suggesting that this could actually have a positive effect. But, and we also essentially did some other studies where we tested multiple doses of this near-infrared light given, as I showed, through a laser, and essentially found that some intensities of the light are much better in terms of triggering biological effects, such as changes in blood flow and changes in the cytochrome C oxidase activity, which are the ways in which we think this laser light could actually affect positively the brain and people's cognition. So on the basis of this preliminary research, we were very fortunate to get funding from both the Alzheimer's Association Part the Cloud program and from the National Institute of Health and the National Institute of Aging to do a very ambitious and relatively large study where we would essentially test in a large group of individuals with mild cognitive impairment or very early forms of Alzheimer's. First of all, to test that this is well tolerated and safe procedures, which we think it is, and then also to test its efficacy in terms of improving people's cognitive abilities over the span of 8 weeks. And we're also doing some biological tests where we use positron emission tomography to look at the amount of tau in people's brains. We also look at changes in mitochondria using an in vivo chemical analysis technique called phosphorus magnetic resonance spectroscopy. And we also look at the acute effects of the laser on people's blood flow in the brain. So not only we want to show that this is a good treatment, but also that it is a treatment that truly works through the mechanism that has been posited. And in this way, even other treatments that work on enhancing mitochondria could be justified for future studies. So just if you're curious, this is what the actual device looks like. So it's essentially just like a bike helmet, a little bit bigger, and there are these probes that are associated, connected with fiber optic cables to the console where laser diodes push these high intensity laser that then goes through the skin and the bone into the brain and triggers the putative, the effects that we discussed. This is how the study actually looks like. So essentially, people do a very complex initial screen which involves neuropsychological testing and positron emission tomography and magnetic resonance spectroscopy, and then they get randomized to an 8-week treatment with either the active photobiomodulation or sham. And there are 3 sessions per week for 8 weeks, so a total of 24 sessions. Each session lasts about 11-12 minutes when you wear that helmet that I just showed you. And then at the end, we do again the neuropsychological testing and the MRS. And then there's going to be another follow-up 3 months later just to see whether the effects are going to continue and whatever positive effects are going to continue over time. Now, this is actually, I want to just highlight all my collaborators that are helping with this study. And it's very exciting because if this would work, actually, this could be eventually a treatment that could be done at home. It would be just like you're buying your exercise machine, you would purchase or rent these types of photobiomodulation devices, and even if the treatment is short-lived and we don't see effects happening 3 months later, it would be not very complicated for people to keep having these treatments for a long period of time. I want to stop here and thank you for your attention. And if you have loved ones or relatives that are suffering from the early signs of this illness, please consider having them join our study. We have 2 locations, one in Midtown New York City, the other one just outside of New York City in Orangeburg, and a 3rd site in Boston, Massachusetts. Thank you so much.
Thank you so much, Dr. Iosifescu. And I just want to make sure that if everybody knows that if they are registered for this program, we'll be sending this information out to registered participants. So thank you so much. Now, Dr. Varga.
Hopefully you guys can see my screen okay.
So
my name is Andrew Varga. I'm an associate professor of medicine at the Icahn School of Medicine at Mount Sinai. And I'm a neurologist by training with fellowship expertise in sleep medicine. And my whole program of research really focuses on sleep and its relationship to neurodegeneration and Alzheimer's disease. So I have a fairly broad research program that spans both basic science and clinical science. My basic science program focuses largely on a mouse model of tauopathy. So this is a mouse that's been genetically bred to have a human form of tau expressed in its brain. And that form of tau has a mutation that makes it more likely to accumulate and form neurofibrillary tangles that Dr. Tavares mentioned in her talk. And as these tangles develop in the brain of this animal model, they develop neurodegeneration that can be seen, I would say, fairly obviously in this picture on the lower left here, where there's a big, essentially, hole in the brain on the right as compared to a normal mouse on the left. And so one of the things that we do with these animals is to monitor sleep. The way we do that is that we surgically implant EEG and EMG electrodes into the brains of these animals, and this lets us measure brain activity and allows us to differentiate things like wakefulness from the different stages of sleep, including non-REM sleep and REM sleep. And we use a drug, something that's called DORA-12, which is a preclinical version of the sleep medicine suvorexant, to try to improve sleep in these animal models. As you might imagine, sleep starts to break down as these animals age, and as a matter of fact, the sleep breaks down before some of the cognitive changes occur. And we have some promising data showing that this drug does in fact rescue many of the sleep deficits in this animal model acutely. And then we're also trying to answer questions about what happens if we modulate sleep bidirectionally early in life. So what happens if we take this animal model, disrupt sleep on a very regular basis for several months early in its life, and then assess later in life what's happening to its cognition and what's happening to the accumulation of this Alzheimer's disease proteins in its brain. And similarly, we ask the opposite question, which is what happens if we augment sleep in some way on a chronic basis in the early life of these animals? And similarly, what's the effect on cognition? What's the effect on accumulation of tau and other proteins in the brain? But I also do a lot of clinical research. So we do sleep studies in human subjects or people. This is my former postdoctoral fellow, Corey Kam, who's now an assistant professor at Mount Sinai, and we collaborate still to this day on a lot of projects. As you may know, one of the earliest cognitive deficits in Alzheimer's disease is actually a deficit in spatial memory or spatial orientation. And this is a little picture from an ad actually for a medicine that's meant to treat Alzheimer's disease, saying that Alzheimer's patients can feel like they're trapped in a maze. And so building on that observation, we actually developed a very specific spatial navigation task that uses a computer platform, essentially, where we have people use a joystick to just kind of navigate through a 3D spatial environment. And so shown on the left is a bird's eye view of this spatial maze that we developed, and shown on the right, it's kind of the first person experience as you're kind of navigating through these corridors and making your left turns and right turns, et cetera, and trying to find a target in this labyrinth. And in addition, we do a lot of collection of fluid biomarkers. So we collect plasma, blood. We also collect spinal fluid. We look for some of these proteins, again, focusing on amyloid and tau, among some others. And then we also do a lot of brain imaging with people. So we do something that's called functional MRI brain imaging, which is a technique that allows us to measure blood flow to different areas in the brain. And it's thought that where the blood flows in the brain is reflective of increased brain activity. And we can do this while people are actually playing this game and navigating in our 3D spatial labyrinth. And so we've been doing some experiments where we take essentially healthy volunteers and disrupt sleep in a variety of ways, either specifically disrupting non-REM sleep, otherwise known as slow wave sleep or SWS, or disrupting REM sleep. And we've shown that there's some really interesting changes in the way the brain processes this spatial information using that technique. And then in addition, we also do positron emission tomography or PET imaging. And this is imaging that allows us to specifically use tracers for amyloid and tau to look at the levels of accumulation of amyloid and tau in the brain. And so just like Dr. Iosifescu, I also collaborate with Ricardo Osorio, who's at NYU, and he's a good friend of mine. And we have a couple studies together, studies in which we focus specifically on aging, and we're interested in understanding sleep as a potential preventative intervention for the development of cognitive decline in Alzheimer's disease. So in some of the work with Dr. Osorio, we focus really specifically on specific brain waves that are seen during sleep. So good, healthy sleep is thought to be characterized in part by these bursts of high frequency activity that are called spindles, highlighted with this SP here in this EEG tracing, as well as by these larger low amplitude or high amplitude, low frequency waves that are called slow waves or SW marked here. And so good sleep is characterized by lots of spindles and lots of slow waves. And interestingly, these markers of sleep increase following learning. And with aging, they tend to decline. They tend to decline somewhat differentially. And we think that the degree to which they decline might end up actually predicting people's cognitive function or their risk for Alzheimer's disease. So a lot of the studies that I work with Ricardo Osorio focus on that. So there are two in particular that we work on together. One is called Sleep, Aging, and Risk for Alzheimer's Disease, or SARA. And the essential inclusion criteria is that you just have to be a cognitively normal older adult, 55 and above. And we have people do all of the testing that I just described, the spatial testing, blood and spinal fluid collection, lots of different brain imaging. We have people do that once at baseline, then again, about 2 to 3 years later to see how some of these measures of sleep end up predicting some of the changes over time. And then we also have a study that's called Sleep Aware that is fairly similar, but focuses very specifically on an African-American population. As Dr. Tavares mentioned in her introduction, African-Americans are at increased risk for Alzheimer's for a variety of reasons. Our interest in this stems from the observation that African-Americans actually have decreased slow wave sleep compared to age-matched non-African-Americans. And we think that may play into part of their increased risk for amyloid accumulation and risk for dementia over time. And so this study is recruiting cognitively normal Black adults over the age of 55. And they're going to do the same types of things: sleep testing, blood and spinal fluid collection, memory testing, and brain imaging over a 2 to 3 year period. Now, one of the things that I'm also very interested in is obstructive sleep apnea, which is the most common sleep disorder and involves essentially repetitive closure of the upper airway during sleep. And so what ends up happening is that people literally stop breathing for short periods of time before they start breathing again. This phenomenon can happen actually many times over the night. In some people in which sleep apnea is severe, this phenomenon occurs every 30 seconds to 1 minute. And it just happens over and over again. And when that happens, obviously levels of oxygen are going down in the body because you're not getting that oxygen coming in. And also your sleep gets very fragmented, and many people with sleep apnea are unable to get into some of the deeper stages of sleep, like slow wave sleep or REM sleep. And there are some treatments for obstructive sleep apnea, things like continuous positive airway pressure, or CPAP, that involves using a mask, or using a dental appliance that helps pull the lower jaw forward and helps to open up that airway. And so we have a number of studies that really focus on assessing sleep apnea as a risk for Alzheimer's disease. So these include a study that's called Slow Wave Sleep and Navigation, or SWAN, in which we're looking for adults of any age, really, that have established sleep apnea and are actually adherent to CPAP therapy on a long-term basis. We have another study that's called Treating Apnea Reducing Tau, or TART, in which we're looking for older adults, 55 and above, that have a new diagnosis of sleep apnea, that are naive to treatment, but are interested in getting treated for the first time, predominantly with CPAP. And then finally, we have a study that we're really just getting started. It's called Preventing Alzheimer's Through Sleep, or PATHS. And it's a somewhat similar idea to TART. We're targeting older adults with a new diagnosis of sleep apnea, but this study is actually a real randomized controlled clinical trial in which people are going to be randomized to either getting treatment essentially by any means, whether it's through PAP, an oral appliance, positional therapy, or some combination of those things, or waiting for a period of 3 months to initiate that treatment. In that 3-month period of time, looking at measures of cognition and changes in blood biomarkers for Alzheimer's disease. So that's really it. That's kind of an overview of the types of studies that I work on. The person here is Ahmed Fakouri, who's my senior research manager, and similarly to what Dr. Iosifescu said, if any of you are interested or know people that may be interested in participating in these studies, please reach out to either Ahmed or me at these emails listed here. Thank you.
Thank you so much, Dr. Varga, for your presentation. And so I want to encourage people to put their questions in chat, but right now I want to go ahead and answer some of the questions that came in through the chat. So one of the questions I saw are, what are the names of the 2 new drugs that are going to be submitted for FDA approval? And we think it likely, based on some of the signs the pharmaceutical companies have made, that the 2 drugs that are likely to be submitted are donanemab and lecanemab. They're also monoclonal antibodies that are, again, a similar mechanism as Aduhelm. And so we think in early part or middle of this year. It's not clear how if those plans will change based on what we know about CMS's medical determination in terms of their initial stance in terms of how they will cover Aduhelm. But that's what we know right now. So one question, which I think is directed to me, is how did we go about picking sites for the US Pointer study? And so the Alzheimer's Association is the primary investigator in that study with other primary investigators at the site. And the decision for the sites was one who could be the co-PI in those sites, making sure that those sites were capable of recruiting a diverse clinical subject base, and also that for the community piece based on exercise and social engagement, that there were also partners in those cities that could partner with the researchers. So it would be a true community-based study looking at those risk factors and the multimodal intervention. So I think this next question is for Dr. Iosifescu. And the question is, what is the latest view on targeting mitophagy as a treatment or possible cure?
To be honest, I'm not the best person to answer this, since I do no research on mitophagy. Essentially, that has to do with the process of autolysis of mitochondria, but we're trying to prevent the mitochondria dysregulation at a much earlier stage with the research that we are doing. So essentially, we're talking about cells that have nearly intact mitochondria in the research that we are able to do with the near infrared light.
All right, thank you. So I think this might be for you, Dr. Iosifescu. They asked about the light therapy treatment. How would it work for people with pacemakers? Would there be an issue to receiving that therapy?
So there wouldn't be any issue for the light therapy. There would be an issue for the MRIs that we do as part of our study, because the kind of high resolution MRIs that we do at 3 Tesla would not allow people to have pacemakers. So we would still allow people with pacemakers in the study, but without doing any of the associated imaging. The actual laser light itself has no impact whatsoever on the pacemakers.
Okay, thank you. Dr. Varga, I have a question for you. What should a regular person know about sleep and the risk for Alzheimer's disease? And do you have some general ideas on what people should be doing while they're waiting for the research in terms of their sleep hygiene?
Sure. I mean, the short answer, right, is that sleep duration is important and quality is important, right? So the first thing is it's important to be getting enough sleep duration, and tons of people really cheat themselves of that all the time. There's a broad range of what's considered quote unquote sort of normal duration that kind of ranges from 6 to 9-ish hours or so. I think there's some gray zones. I mean, I think certainly people that get 6 hours of sleep on a day in and day out basis and aren't catching up, for example, on the weekends, probably are having some deficits cognitively or otherwise, and let's say maybe increasing the risk for all sorts of negative health outcomes. So like I said, focusing on just sort of getting enough sleep, I think, is one aspect of it. And then the other one is the quality, right? So I highlighted sleep apnea, which is, I think the most common sleep disorder, but insomnia is also extraordinarily common. And so really anything that people identify that's disrupting their sleep, whether they're able to identify it themselves or whether a bed partner is able to identify it, I think are, important things not to ignore and important to potentially seek out, a sleep physician and get addressed.
All right, thank you so much. I'm going to give one last chance for people in the chat if they have questions. We're almost at time, and I'll be able to take one more question. So you can type things in the chat. It's at the bottom of your screen. And I will wait to see if anything else comes in. If not, okay, so a new question came in for you, Dr. Varga, and the question is, if you get 6 hours of sleep, but it's because you wake up, not deprive yourself, does that still lead to deficits and increased risk?
Yeah, so it's a little hard to answer that question because truthfully, that's actually fairly rare, right? So there are genetically short sleepers, but they're a very small part of the population. And so people that are really sleeping 6 hours and not getting any catch-up sleep on a day in and day out basis, like I said, they either fall into one of two categories, which is they either are part of that rare subset of people who are genetically predisposed to short sleep, or they really could be getting more sleep behaviorally or otherwise. So I guess that's the short answer.
Okay. Well, thank you so much for your wonderful presentations and this conversation. I'm getting through the chat that people are saying that this is such an informative session and they really enjoyed it. So I thank you so much, Dr. Iosifescu and Dr. Varga. As I said before, this is being recorded. We will share this on Facebook. We will be sharing links to TrialMatch, these research studies, for any of you that are interested. And we encourage you to share the information that you received today with your friends and family members. Again, we will be having another session, but we often have talks at the chapter level, introducing you to research. So again, I encourage you to take a look at TrialMatch and downloading the Science Hub app, because you can have all up to the minute and credible research information in that science app. So thank you so much for spending your lunchtime with us. If you have any questions, want to learn about additional programs, you can learn more on our website. Call 1-800-272-3900. Again, thank you so much for attending.
Thank you.
Thank you.